Hello, everyone. Thanks so much for joining us this morning for Ask Me, our educational webinar series. My name's Audra Hines, Vice President of Care Services for the ALS Association Golden West Chapter. I want to give a very big thanks to our generous sponsors, Dwayne Morris, Attorneys at Law, Biogen, Gladstone Institutes, Brainstorm Cell Therapeutics, the Center for Neurologic Study, Alexion, Cytokinetics, Novartis, Mitsubishi Tanabe Pharma America, and the Barber family. We absolutely could not make programs like this happen without your support, so thank you so much. And just a quick reminder that all of our programs and support groups remain virtual at this time. Please go to www.alsagoldenwest.org to check out our lineup of all upcoming groups and events. As always, these are free of charge and everyone in our communities are always welcome. I'd now like to turn it over to the Golden West Chapter President and CEO, Fred Fisher, for a few remarks and to introduce our first speaker. Fred?
Thanks, Audra, and good morning, everyone. It's so great that you can join us, and we're all really excited for you to get to hear the latest and greatest research. We've finished 2 full days of scientists connecting with each other, talking to each other, presenting to each other, and challenging each other around new concepts, new ideas, and updates on some really fascinating research. So we're excited for you to hear all of that today from our presenters. I want to just give this opportunity to you to understand that at the Golden West Chapter, everything we do advances the search for effective treatments and cures. So when you support the chapter, either through the Walk to Defeat ALS, the Ride to Defeat ALS, our campaign for cares and cures, you are advancing and accelerating the search for effective treatments and cures. And I just want to spend a minute describing to you how that happens. Everything we do at the Golden West Chapter is really focused on that top center circle. It focuses on people with ALS and their families. When you engage with us through our community-based care services, we want to make sure you're getting access to the best possible care and that you have access to all kinds of opportunities, including advocacy and awareness. When you tell your story, it's that story that is the most compelling motivation for our elected representatives and your friends and family members and coworkers and employers to support the mission of the Golden West Chapter. And that money that is generated from those advocacy, awareness, and fundraising efforts, that money goes into funding for government funded research and privately funded research. And that money goes into labs where great ideas are developed. Now, cures aren't found in labs, but ideas are developed in labs. And in order for an idea to become a treatment or cure, it's got to go into one of our multidisciplinary ALS clinics. And we're so fortunate here in California, we have 8 certified treatment centers of excellence. These are the places where labs, biotech companies, pharmaceutical companies go to have their ideas tested on people living with ALS. And of course, that brings us full circle, that everything we do centers around people with ALS and their families. And you can see if you took any one of these pieces out, if you took the people out, if you took the service and outreach out, if you took the advocacy out, if you took the funding out, if you took the labs out, if you took the clinics out, any one of those pieces comes out, the system falls apart. And it's why we at the ALS Association and the Golden West Chapter in particular has advanced all of these efforts. It's why we need all of these efforts working in harmony to assure that we are moving as quickly as possible with urgency to find treatments and cures for ALS. And it's why we're committed to all these things. So when someone says, why don't you put just all your money into the science? It's because we need these other components working. together with the science to make sure we're developing the best treatments and cures for people with ALS. So when we talk about funding for science, what we have a whole division at the ALS Association focused on funding science. And the head of that division is Kuldeep Dave, who is the vice president for research and joined the association in May of 2019. In his role, he develops and implements the broader research vision by setting annual priorities and department goals, including setting the research strategy for funding, for what programs are going to get funded, and what strategic initiatives we're going to move forward as the ALS Association. He's also responsible for the operational staffing and budgetary oversight of the research program. A little background on Kuldeep. He received his bachelor's degree in biology from Rutgers University. He received his PhD in pharmacology and physiology from Drexel University College of Medicine in Philadelphia. And he went on to work in the pharmaceutical industry before joining the Michael J. Fox Foundation for Parkinson's Research. So he is no stranger to neurodegenerative disease research. And after 9 years at Fox, he joined the ALS Association as the head of our research program. So it's my pleasure now to introduce you to the Vice President of Research for the ALS Association, Dr. Kuldeep Dave.
Thank you, Fred, for the invitation, and I wish I was there in person to talk to you all. I'm going to start sharing my slides, and hopefully it will work. Please let me know when these come up.
Yes, they're up.
Okay, great. Give me 1 second here. Okay, so thank you again, and thanks for that introduction, Fred. This was a great conference last couple of days, and I'm happy to provide a very, very high level summary on what our program is. And as it says up here, we are the largest philanthropic funder of ALS research in the world. And just some of the key things to keep in mind about our program. First of all, as you can see, there are arrows, and this is really supporting the point that Fred was just making. Research cannot happen in its own, on its own. And what makes our organization so great is that our mission is a three-pronged mission: care services, advocacy, and research. And they work in tandem with each other and inform each other and each other's strategy. So within research itself, right here in the gray section, since the Ice Bucket Challenge, we have committed over $110 million directly into ALS science. We fund academic labs, and we fund pharma and biotech companies, and we fund across the world. And this is different from other organizations. There might be other nonprofits that only fund academia or only fund a particular area of the world, or maybe their particular state. We believe that good ideas come from everywhere, and we fund all across the world. We fund research projects, and I'll go into this, sort of detail on where we, what kind of research we fund and what type of projects. But we also fund infrastructure projects. This is what keeps research going. Things like clinical trial infrastructure, or databases to share data across the world, or biospecimen sharing so that people have access to biofluids or biospecimens to do research. We also fund training and education grants. It's important to have new scientists keep coming into the ALS space and staying in the ALS space. And so we fund fellowships both at the preclinical research and at clinical fellowships to, again, keep that new brain power coming into ALS research. And I think what makes this difference is the scope, the magnitude, and this integration of mission, which is key, and it sets us apart from any other ALS organization. And finally, the last point, we may be the largest philanthropic funder, but we're not the largest funder. We fund about, somewhere between 15 and $20 million each year. But the NIH, the National Institutes of Health, funds over $100 million. So they're funding five to six times more ALS research than we do. And so our goal, our sweet spot, is being able to leverage our funding. And what we found out last year when we did an independent study, that our $40 million of our funding was able to then allow researchers to get $120 million of follow-on funding. And this is our leverage. We can leverage 3 times of our funding, and that, I think, is our sweet spot. Okay, so before I talk about the various projects, I think it's important to understand the research ecosystem. What does the ALS ecosystem look like? And so I'll start all the way up here in the left side in red, where it says biology and genetics of ALS. This is really important first key step. This is what I think Fred mentioned happens mostly in academia. We need to understand what is different in ALS biologically, so that we can then make drugs against that biology. And so this is what we call, this is the area where we do gene sequencing, genetics, try to understand what may be different in ALS and what pathways are different, what mechanisms are changed. That all of that is happening in that red portion, biology and genetics. And underneath that is what are the risks and causes of ALS? And this is beyond genetic risk. What are the environmental risks? What are the occupational risks? We know that people who serve in the military have a double risk of getting ALS. What are we exposed to outside of our genetics, outside of our physiology, that may increase our risk of ALS? Once you have that, you then move to this next area in green called drug development and biomarkers development. And this is where now that you understand the biology and you know something is different, now you make drugs against that biology to try to rescue or recover the ALS pathology. And so this happens normally in pharma and biotech. This is where you do chemistry, or you make biological drugs, you do lead optimization, screening, testing in animals. You test whether the drug is safe in animals, it works in animals, and biomarkers development. These are objective markers that tell us about the disease and whether the drug is working. And once this happens, and you know that you have a drug that works preclinically, is then when you go into this next phase in blue, phase 1, phase 2, phase 3. These are 3 different phases in the clinic. Phase 1, you test whether the drug is safe in a smaller ALS population. Phase 2, whether you now, again, still in test for safety, but now you start to ask questions on efficacy and whether it works in a smaller ALS population. And then phase 3, where you again test for safety, but now you start to look at efficacy in a larger population. So that's the drug development pipeline. On the other side of it, in green, is research for people living with ALS today. So that's things like phase 4 studies, which is how are PALs, the medications that they take, how effective are they? Research into assistive technology. Technology is amazing. It's we are able to have our phones tell everything about our life, and can we use technology to make better wheelchairs, better DME equipment? Research into telehealth, telemedicine, especially important in this time of COVID. What is the burden on families and caregivers? And there's research that can be done into that. Access to care, quality of care, natural history of the disease, and surveys asking the patient about the disease. And then underneath that all you can see is the infrastructure piece, which is very important because all of this stuff in the top cannot happen without the infrastructure, without data sharing, without biospecimen sharing, without tools being developed in the lab, training for fellows and scientists. So again, all of this is very important. And what you see here is the ALS Association funds almost has a very wide footprint. We fund almost everything here. Even when we, as with most nonprofits, we cannot fund phase 3 studies because these cost over $50 million each. We can still fund a small part of that, like biomarker type studies and companion studies, which help understand the results of phase 3. And I'll talk about this in the next slide. So again, the take home messages here are our program is wide, large footprint, scope, a fairly wide scope. And just to give you a sense of what we're funding now, 175 active projects we have currently spread across 15 different countries in the world. And if you count up the amount of money that we've committed to these 175 projects, that adds up to a $58 million research commitment that we have to those projects. So Fred wanted me to give you a taste of the type of projects we have funded. So in the last 5 minutes, that's what I'll do here. So, 2020 is a hard time. COVID hit, and not only did it impact people and our lives, but it impacted research in a great way, both because researchers could not get in and do research in the labs because of social distancing, because of closures, but also how COVID impacted fundraising across the nonprofit space. And despite those limitations, we were able to commit over $14 million to 50 different projects last year. So let me just walk you through some of these projects that we funded. So starting on the top here in left in blue, $3 million to the Healy ALS platform trial. And I think you all heard about the platform trial yesterday in the session, the clinical session we had. This is a very innovative way to do clinical trials where you can run multiple drugs all at the same time. So you're not running one clinical trial, taking the infrastructure down, and then putting another clinical trial up, and then putting the infrastructure down. If you run a platform trial where you can run 4, 5, 6 drugs all at the same time, and they all share a placebo, and you leverage the common infrastructure, you save time, you save money, and we were very happy to provide $3 million to that program. If you go down one to that red area, we provided $2.5 million last year to 5 different preclinical drug discovery projects. So remember, I was telling you, you get preclinical drug development and then clinical drug development. Healy is the clinical drug development. The preclinical drug development, we provided $2.5 million. And in fact, 2 out of those 5 projects are in California that we funded for preclinical drug discovery. We provided $650,000 to NEALS. NEALS is a consortium of investigators and sites across the United States. And frankly, no clinical trial would be possible without NEALS infrastructure, providing that kind of knowledge and know-how and expertise for running clinical trials. And we were happy to provide funding to NEALS directly for that type of infrastructure. As I told you, we also fund research that helps betters the lives of people living with ALS today. And so we funded 7 projects to the tune of $1.4 million to provide for research into technology, quality of life, better assistive technology, so that we can have an impact on this research can have an impact on people's lives today. I talked to you about phase 3 trials and how our inability to fund a full phase 3 trial, but we can fund companion studies. And we in fact funded, and this was in collaboration with IMALS, a 500K study for biomarker measurement for the Neuron Brainstorms phase 3 trial for Neuron, which is now turning out to be an extremely important piece of data in their study. Partnerships are very important. So again, IMALS partnership for the Neuron trial, with partnership with Target ALS, which is another nonprofit organization. We funded an assay, a TDP43 assay. TDP43 is a protein that is the main pathological protein in ALS. It's the protein that goes dysfunctional, and you see pathology in the brain and spinal cord. And we, in partnership with Target ALS, gave a grant to develop a biomarker assay so we can measure this protein in biofluids. Going up to the red in the right, I told you it's important to keep having new people coming into ALS space. And we know that if we can give them funding at early stages, then they're going to stay in that space. And so we funded 4 preclinical and 4 clinical fellowships so that those young fellows we can seed research and they can continue on that path in ALS research. This in green, we gave again a partnership with Project ALS. This is another nonprofit organization. And we provided $650,000 for developing and testing a new gene therapy approach, the antisense approach, against this genetic protein mutation FUS to Dr. Neil Schneider at Columbia University. And finally, here in purple, we launched last year ALS Focus, which is a patient and caregiver survey program where we want to hear from you. We want to know how the disease is progressing, how whether it's quality of care, whether it's insurance, whether it's caregiver and their burden. That's a program that allows us to hear from you directly. So I'll stop here, and because my 15 minutes are up, happy to take questions now. And I hope that this gave you a good sense of the type of work that we fund. Thank you.
It definitely did, Dr. Dave. Thank you so much. Lots of wonderful information there. We do have a couple of questions. Wanted to ask first, is there an update on compassionate use approval of Amylyx?
Yeah, so just maybe bringing everybody to speed on this. So Amylyx is a company that reported on their Centaur trial. This was for the compound AMX35. And so the question is asked, and a couple of things to add to that. One, the ALS Association supported this trial through Ice Bucket funds back in 2016. And second point, even more important, is that Amylyx reported positive data both on function and survival, as well as safety in this study. And so we're really excited. As you know, we led a petition for the FDA to approve this compound as soon as possible. We do not have information on the compassionate use or expanded access, I think is what the question is. On Amylyx, I know the drug company is thinking about this and, sort of how this relates to their drug approval and access. But unfortunately, I don't have any more information than that.
Okay, great. Couple more we have here. Being in the military, you're twice as likely to get ALS. Have you drawn any conclusions or educated guesses of what the possible causes of sporadic ALS are?
Yeah, very good question. So there have been a couple of studies that have looked at, what may be happening during military exposure. Is it exposure to, weapon grade weapons? Is it chemicals? Is it that you're out in the field and, there's training, and that training is, stressful. So is it exercise? And I think it's been difficult to pinpoint a single factor. This is why the VA and their research arm becomes very important, because they actually have samples that they provide to researchers. By the way, this also happens through the CDC registry samples as well, that provide samples to try to understand what it is in someone's military experience, what exposures is it in their biofluids that may give us some inkling about, what changes. Just to one quick point on this, we at the ALS Association are launching a prevention initiative, and the idea is exactly that, which is, can we identify these markers early on and prevent and provide measures and guidance to the community on, what can they do to lower incidence of ALS.
Wonderful. Last question for now. A couple of people had questions around familial ALS and wondering if there are any promising drugs for that type of ALS.
Oh yes. So so again, let me back up because I'm not sure everybody understands what familial means. So when you look at ALS and you try to figure out how many people can you explain may have gotten ALS because it runs in their family or it's genetic, familial, versus people where it may not be related to any particular gene. And so if you look at that, that's 90-10. 90% is sporadic, where we can't explain it, and there could be many different factors. And 10% roughly is genetic or familial, as you say, Audra. And so again, I want to make sure everybody understands that before I answer the question. We have learned an awful lot about genetics in the last 5 years. We now have over 20 genes that we have implicated. This is a lot through our efforts, our funding of gene sequencing approaches like Answer ALS, Project Mine, Create Consortium, New York Genome Center. I mean, we have funded huge gene sequencing approaches so that we can identify those genes, and those genes have been identified. And happy to say that there are several approaches now in the clinic. For example, Biogen has a phase 3 trial currently for a drug called tofersen that is targeting SOD1, which is one of the major genes that provides familial, that sort of provides that familial or contributes to the familial ALS. And so I know that there are other companies that are developing compound genetic approaches against C9 or 72, and it's not just in any sense approach like Biogen has, but AAV approaches as well. You heard from CRISPR yesterday, and Clive's going to talk about that later today. But again, the point is that a lot has been learned, and we've made a lot of progress on the genetic side.
Wonderful. I know we have a few other questions, but I'm actually going to save those for some of our other panelists. So thank you so much, Dr. Dave, for being here. Wonderful information. I know that our audience really wanted to hear. So much appreciate your time.
Thank you. Always happy to do this, and appreciate it. Have a good conference. Thank you, everyone.
Thank you so much. I'm going to turn it back to Fred Fisher to introduce Dr. Clive Svenson.
Thanks, Kuldeep, for a great presentation overview of the work that the ALS Association is doing. You can see we're covering a lot of bases on the research front. And just to connect the dots to that last question, it was the ALS Association that funded the proof of concept that antisense technology could be effective in the treatment of genetic forms of ALS. And so that tofersen drug that now Biogen is moving forward in a phase 3 trial, that actually started with funding by the ALS Association to determine whether or not that technology could work in ALS. So it's great that we can see the the connection between your support of our efforts and that support resulting in real therapies that show real promise. Turning now to Dr. Clive Svenson. Clive did his pre-doctoral training at Harvard University and received his PhD from Cambridge in England, where he established a stem cell research group before moving to the University of Wisconsin in 2000 to become the professor of neurology and anatomy and director of an NIH-funded stem cell training program and co-director of the university's Stem Cell and Regenerative Medicine Center. In 2010, we were fortunate to be able to lure him to Los Angeles and establish and direct the Cedars-Sinai Regenerative Medicine Institute, which currently has 15 faculty members and approximately 100 staff. The focus of their research is to derive cells from people with ALS and other disorders, which can then be reprogrammed to create powerful models of human disease. Dr. Svenson led the first groups to successfully model both spinal muscular atrophy and more recently Huntington's disease using this technology. And the other side of his research involves cutting edge clinical trials. He's involved with one of the first growth factor treatments for Parkinson's disease and is currently working closely with neurosurgeons, neurologists, and other scientists to develop novel ways of using stem cells modified to release powerful growth hormones to treat patients with neurological diseases such as ALS, Huntington's, Alzheimer's, and Parkinson's. Clive is also the chair of the California ALS Research Network, which works collaboratively with the Golden West chapter to produce our annual research summit. And so we're really fortunate to have Clive here with us to talk about what happened over the last couple of days, including some really interesting research looking directly at those environmental factors that may cause ALS. And with that, I'll turn it over to Clive. Thanks for being here.
Fred, thanks very much. Just a microphone check. Can you hear me okay?
Yep, hear you good.
Great. Well, lovely introduction. I think this is our 11th year now that we've been doing this in association with the Golden West chapter. And, you know, a little different this year with COVID, but I'm so glad to see so many people online listening. And we were determined to go ahead with this meeting because COVID is raging, but ALS is still raging as well. And we wanted to really make the point that we have not stopped our efforts this year because of COVID. And in fact, we're doubling down. There's a lot of excitement. A lot of the things Kuldip was just mentioning, we were discussing over the last 2 days. The next slide. This meeting is really about collaboration. It's really about unique in a sense that we get not just the researchers, but we also get the industrial folks, all the companies working in this space, and we get the clinicians, and we all sit in the same room usually, as you can see in this photo. This time we sat in our Zoom room to try and learn more about ALS and very specifically the West Coast and California and how we can collaborate together and work together in many times with the ALS Association through initiatives like the Neuro Collaborative, which brought together 3 groups in California to do some groundbreaking work, which has now resulted in some interesting developments that I'll tell you about in a minute. So a lot of the investments we've made have really paid off. Final thing is this summit is for the patients as well. This is why we do this. After we meet and discuss all the nerdy science stuff, we want to disseminate that to everybody. You're the reason we do this. So this is why we have this particular meeting right after the summit and maintain and retain some of the amazing speakers that have just given their presentation so that we can, you know, give this to you guys and give you a chance to respond, because it's all about the patients, and that's why we're doing all this work. So this is now a mission just that I just described. The next slide is another more recent summit. This year, you know, when I started this with Jim Barber and Fred 11 years ago, we really wanted to keep this small maybe 50 people or 60 people. And we managed to do that successfully many years. We never advertise this outside of California. It's just the group, you know, to get together and with industry. It just keeps growing, which is a good sign organically. And this year we had 260 summit attendees through Zoom. Of course, we're not restricted by flying people all over the place, which is wonderful. So we actually had double our normal group number of people. We had 50 guest speakers. And this time we actually had a good colleague of mine from Tokyo who's doing, Hideyuki Okano, who's doing some amazing work on ALS from Tokyo. And we learned a lot from him in his presentation. Now the next slide. We've had 2 Nobel laureates talk at this. The first one was Stan Prusner, who talked about 8 years ago. And we're very lucky to be able to get Jennifer Doudna to talk this year, who just won the Nobel Prize in Chemistry for her work on CRISPR. And to describe that, I'll let you do it because I'm not as good at doing that as she is. I think we have a short video that they're now going to tell, that they're going to show Jennifer and her work. So I don't know if you want to play that video now. And this is very relevant for ALS because what she's going to talk about is editing, CRISPR editing, which means just what Kuldip just mentioned. If you have a genetic form of ALS, this technology may be able to go in and repair that gene deficit in the future and reverse the cause of the ALS. So if you play the video, please enjoy this.
I think the thing that's amazing about science, biology for me, is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.
Well, just fantastic stuff. You know, the Nobel Prize this year, and Jen is just one of the most nicest people. We had a great discussion with her. She is very interested in ALS and how she can apply that technology. You'll hear from Steve Finkbeiner later, and her lab, one of her labs is at the Gladstone, is right below his, and they're already collaborating on ALS. And she was telling us about some of the clinical work they're doing, in particular in another disease called sickle cell anemia, where they've actually used it in patients in sickle cell, and they're having some success with this technology. So the idea is just to find out what your mutation is, what's causing your ALS, and then we can use this technology, put these CRISPR elements into your bloodstream, and then they'll go into your cells and repair your DNA. It sounds like science fiction, but it's just around the corner, and she was an amazing addition this year. So from her to you guys, she really wanted to be here today, couldn't make it. When you get the Nobel Prize, you tend to get pretty busy. So we were incredibly lucky to get her yesterday, on Thursday rather, and she sends her best to all of you, and I just pass that message on. So look, this is a point often in years, I'll spend 10 or 15 minutes running through everything that happened, but I think Caldit gave you a very good overview, and I really want to get to the questions. So I think I'll go through some of the flow of what we discussed at the meeting, and then I'm seeing lots of questions come up, so I think I'll get into the questions pretty quick. We had usually a diverse set of topics. The way we do this meeting is we all meet up like 4 or 5 times before the meeting to discuss what's the most exciting part of ALS this year. We usually leave it until a few months actually before the meeting so that we get the latest areas that we're interested in. So obviously gene therapy with Jennifer Doudna, excuse me, talking about this was a lead-in, and then we had some remarkable speakers showing not only the excitement of this area, but how can we really get these technologies through into the clinic. We had someone called Chris Bankovich, who's a leading neurosurgeon at UCSF, who's actually developing these technologies that you can infuse these CRISPR elements or other elements into the brain using a particular type of cannula and very, very focused delivery into different areas of the brain. We heard from Lucy Bruin, who you all know from, who's now at Avexis, about their clinical trials in ALS. And we heard from a panel, including Brian Kaspar, who is really one of the main developers of gene therapy approaches. So it was a very interesting discussion, and I think led to some new ideas in collaboration with people like Don Cleveland for how we can push these gene therapies, whether it's CRISPR or other areas. Another company in the space is Ionis that you just heard about from Caldit, and they're using a different way of editing your genes, but they're using what's called an antisense technology. But how all these technologies can be delivered to patients with ALS. And so I think we came up with some good ideas. We got more collaborations going through those discussions. And we ended very excited on that session with potential new projects going forward for that group of patients that has ALS with a genetic mutation. Now, of course, we'll get to that later. That leaves what we call sporadic ALS, where you don't have a mutation, and that's 90% of patients. However, I think I'm very confident that if we can understand how to treat ALS when you have an SOD1 mutation or a C9 mutation, if we manage to do it for those small group of patients, once we understand what causes the rest of ALS, which we'll get to right at the end here, we can then develop gene therapy approaches, whether it's CRISPR or other approaches, to treat sporadic ALS. We just need to understand what causes it, and if it's genetic, I think these lead trials will give us that pathway, if you like, for treating these sporadic cases. I'm not going to go through in detail all the sessions because of time. I see it's 10:47, and I do want to get to the questions. But we had a session on digital technologies, how they're changing, particularly speech recognition and things that we can do to help patients speak better. And that was also in the big data section. We had people from Google talking about new speech technologies, using recordings from before you got ALS, sort of blending that with your current voice to enhance, or just enhancing your current voice if you have difficulties to make it more audible. We had the poster presentations. I'll tell you about that in a moment. Clinical trial updates. Actually, that was me. We have a stem cell trial that we've just completed at Cedars-Sinai with very encouraging preliminary data. That was a safety trial. So we've shown that stem cell approach was safe, where we put these stem cells in the spinal cord. And we're now moving, if it hadn't been for COVID, we would have done some more patients going a little earlier in the disease and a little different location in the spinal cord. And that's looking very encouraging. And we had an update, of course, from a Neurone brainstorm. And many of you heard that their data wasn't as encouraging as they'd hoped, but they have, in that they didn't get a major effect. But when they looked deeper in the data, there were definitely things happening to some patients, some responding patients, that correlated, which means it was associated with changes to things in the spinal cord and CSF, which Caldit just mentioned. Actually, the ALS Association funded that part of the study, so they may get a lot more out of that trial. But I have to say, it wasn't the outcome, obviously, we all wanted, which was a very positive result. But we're going to learn a lot. Like all these stem cell trials, you're going to learn a lot of information every time you do them. The rest were cryptic exons. This is looking at how TDP43 kills cells, and that's, as you mentioned, Caldit mentioned, is central to a lot of ALS problems. A lovely session on immune system. It turns out in some models, the microbiome in your stomach may affect the brain. And so we had Kevin Egan from Harvard talking about some of his work, and Bob Balow, who some of you remember. Updates from industry, we had a few sessions on the latest clinical trials, including the Brainstorm trial, and in particular the AMLEX trial. That's the final thing I'll talk about, which Kuldeep mentioned as well. Now, this trial was run by, I was very involved with this in the early days because the ALS Association and ALS Finding a Cure all collaborated together to fund them. It's two young boys, basically. They were in their 20s and had this crazy idea that you could use an antioxidant together with another drug in combination. And they were funded, they did the trial, and it's actually looking very encouraging. That seems to have slowed down progression. But, and here's the but that Kuldeep just mentioned, this is a phase 2 study. And unfortunately in ALS, there are many phase 2 studies that seem to have an effect, but then when you go to the bigger study called a phase 3, where you have more thousands of patients maybe, or up to 1,000 patients, many of these promising drugs at phase 2 have failed. And that's because phase 2 isn't a large enough study to really see if this works across many patients. And as you heard, and as they told us yesterday, they are discussing this with the FDA, trying to get different use for this drug because it looked very positive in the phase 2. But the traditional way for these medicines is they will go through a larger phase 3 trial, which they maybe will have to do in order to make sure it really works across a broad spectrum of patients, and that it wasn't just something about that group of patients they used that made it work. The clinicians love to talk about trials. We talked about the design of the trial as well. And finally, the big data and genomics. We talked about Answer ALS, where we're doing 1,000 IPS lines, stem cell lines from patients, and trying to understand, just to come back to my initial thought on genetics, this Answer ALS, and you can go look it up on the website, answerals.org.com. Answer ALS is one of the largest projects collecting information from ALS patients across the country. And that whole Answer ALS project might end up finding new groups of patients with genetic mutations or molecular differences that we can now target with specific drugs. That's the goal of Answer ALS. It's a huge project. I'm very involved with it with Jeff Rothstein and Merit Chakovich, the three of us who are kind of coordinating it. And we're very hopeful the next year or two that'll start coming out with some very good results. And big data means big analysis. And we're working with Google and IBM and other folks to try and understand what these subgroups are. And you may find yourself in one of these subgroups, and we may then be able to develop novel therapies based on that. So I'll stop there and see where I'm going now. I think questions. Yes, let's do questions.
Yes, that would be great. Thank you so much, Dr. Svenson. We have lots of really good questions. We've had a few around Neurone, asking about when that might become available to the public, just status of limited approval.
So Neurone is in that stage now where they've done the phase 3. And the phase 3, unfortunately, wasn't a slam dunk. So, and what I mean by that is it didn't meet its endpoints. And so, unfortunately, that isn't going to translate. Now, if they'd been like a Deveron and their phase 3, and which was done in Japan, met their endpoints, then they put to the FDA for approval, right? So with Neurone, unfortunately, they didn't meet the endpoint that they set. And so my understanding is that's not going to be approved right away. But like the same as for the AMLEX, which is at the phase 2 stage, we don't know where they're going to go next, whether they go to a phase 3, and if that works, they get approval, or they might push for direct approval because it was quite a powerful effect. So the Neurone is now in the position where can they give it expanded use? Can they find other ways to get it to patients? And they're still discussing that. There's no details yet on whether that'll happen.
Okay, thank you for that. Interesting question. Why won't companies like Biogen participate in Right to Try or Compassionate Care for Tofersen or other phase 3 success stories?
You know, this is a very long answer, and I'm not going to give the long answer. There's been many discussions on this. When you're a company, you really want to get that FDA approval. I mean, that makes the whole pathway very straightforward. If you go to expanded use, it gets very complicated because you haven't. So here's a very simple, I'll stop after this. It's very simple. If you haven't proved your therapy has worked in a phase 3 trial, which is the gold standard, you hear it for COVID as well, you have to do a phase 3 trial to prove that your vaccine worked. If you haven't proved it's worked, is it ethical to sell it to patients, even if they want to pay for it? And that's where companies are. They don't want to give you something if they're not sure it's worked. Right. If it's worked, you'll get FDA approval and everybody will be able to have access.
Great, thank you so much. One person is asking, how do we get involved in CRISPR trial?
No CRISPR trials yet. Jennifer's doing it. They're starting in, she told us some nice data that they're doing for hemophilia or other diseases of the blood, sickle cell anemia, but no, nothing for ALS yet, but there are companies working very similarly, as you heard from Coldit, on other approaches for genetic modification of cells in ALS for SOD1 patients or for C9 patients currently. And I think we should give a list of folks, Audra, afterwards. We can give a list of what clinical trials are currently open and available. We should be able to get that through the ALS Association.
Yeah, absolutely. We can do that. Can you discuss, here's another question. Can you discuss current understanding of any immune and inflammatory factors in ALS?
Well, that's a very interesting discussion. We know that the immune system is inflamed in ALS because when you look in the brain or in the spinal cord, you get inflammation. Just like if you get a cold or you get the flu, there's inflammation where the immune system is reacting to something. And so there is definitely inflammation in ALS. You're losing your motor neurons, so you're getting paralyzed and you're getting inflamed. Mm-hmm. The real question, which I don't think has been answered yet, is whether the inflammation is causing some of this and this, or whether it's a result of your neurons dying. And we're still fighting to understand more about that. I think they're, interestingly, you could probably modulate the immune system like Stan Appel has by putting stem cells into the blood, which calm the immune system down and slow down progression perhaps. And there are some interesting clinical trials going on there right now from Stan Appel to see if that will work in patients. But we don't really know the relationship well enough, and there's lots of studies, research going on there to try and understand that inflammatory response better.
Great, thank you. That answers a couple of our questions. Any info on how to improve tolerance of TUDCA, as many people have a lot of difficulty taking it?
This is for the Amilex drug, right? One of the components. No, I have no clue. And again, I have to stand back because I'll say I'm not an MD, so I can't ask those sort of clinical questions. But they're very clever guys. They were great yesterday, and they will be coming up with ideas of how to get over some of the side effects from one of those components for the Amilex trial. But I don't have any ideas on that. Okay,
great. One last question, Dr. Svenson, before I know you're moving on to another video, another part of this section. Someone says, I'm in a clinical trial for C9 using ASO therapy. Can you describe how you see neuroprotective or regrowth drugs working in tandem with gene therapies? Will we need a cocktail approach for a full cure?
I think eventually there'll be cocktail approaches, and we shouldn't be scared of thinking about that. Now, the FDA hates cocktail approaches because when you give one drug, we don't know how another one will interact with it. So it's complicated enough to get one drug into a patient, but when you add another one on, it could do something to the first one, could make it better, or it could make it worse, right? So these trials are quite complex, but I think we have to be a little more aggressive there in terms of trying, now for the Amilex trial, remember, many patients were already on riluzole or even edaravone as well. So those are the two approved drugs. So when you go into a clinical trial, they won't stop your current approved drugs. So in a way, it is a combination trial because you're getting edaravone, you're probably getting riluzole, and you're probably taking lots of vitamins, and you're probably doing lots of stuff. And now on top of that, you're getting the Amilex drug. So I think we are sort of combining things in some senses, but designing those combined trials is kind of tough. Just one last thing, I'll finish on our trial at Cedars-Sinai, where we're transplanting stem cells into the spinal cord and in an effort to slow down motor neuron degeneration. They release factors. We're going to try and do a combined study where we put them in the spinal cord, but we also put them in the motor cortex, because there's two motor neurons that die in ALS. The upper motor neuron that sends a signal to move down to your spinal cord, and then a second motor neuron that goes out to your muscle. And so you need to protect probably both of those, because to have ALS, you have to have damage to both the top motor neuron and the bottom one. Mm-hmm. And there may be drugs that act differently on the upper motor neuron and lower, so we may need to combine drugs to protect both. And I think those are things that we talk about a lot and that we'd love to push towards more combined therapies. Yeah.
I see. Great. Thank you so much. That's actually very helpful. I'm going to save, I know some of you have asked some more questions. I'm going to save these just a minute. I think, Dr. Svenson, maybe we're moving into another video.
I think we are. So I think now we're transitioning to, I have a couple more slides, and then I think I'm going to have my favorite video, which is to do with Jim Barber. And Jim Barber was really the founder of this meeting. He was a patient with ALS who really rallied both the government with Fred and researchers, to be honest, like me, and got us all ramped up and motivated, because he is the true motivator, which resulted in this award, which is called the Barber ALS Research Award. And this has been now, remember, the third or fourth time you've had it. Scientists they don't make much money. We don't go to science to make money generally. And so this award of 1,000 for these young postdocs, and postdoc means you're a student. Now, science is a long job, guys. You have to understand, when you get your bachelor's degree, a lot of people stop there. But when you go into my field, you then have 5 years more to get your PhD, which is another 5 years of no money, because they don't pay PhD students anything, seriously. So then you get your PhD, and all your buddies are going off to do stuff where they make lots of money, and you've still got no money. And then you do your postdoc. So postdoctoral means another 5 years after you finish your PhD studies, you've got to do another 5 years of a postdoc, which means post-PhD. Now, this is where you become more independent, and you start generating data on your own a bit more with a supervisor. And that period is really critical for scientists, because when they get to the end of that, they have a 5-year window. They then have to get their first academic position called assistant professor. But there's 10 years from when you're 20. So you're 30 years old then, and you're still in the education mode. So these kind of awards are really important. That's my way of saying, for these postdocs. And they've really stimulated the postdocs that we've had come through. And these postdocs are the engine of research and clinical trials. Without the postdocs, this ain't going to happen, guys. I don't do much, to be honest. I sit and I give these lovely talks, but it's my postdocs in the lab who are right now, I went in last night, are slaving away because of their love for the research and their passion for ALS to do the work. So that's my shout out for the postdocs. And Jim Barber understood this, and Cheryl, bless her heart, who's on the phone here, has continued this tradition since Jim passed away. And I think what we're going to do now is see a video of Jim, because it's so inspiring, and it will really explain more for those who didn't know him. This is a great way for you to see a little bit about Jim, and then I'll come back after the video and tell you a bit more about the award. And you're going to be lucky, because I'm going to do what I usually do at posters to postdocs, but you'll see it live. I think the idea is to give you a feel for what goes on at the meeting.
Good morning, and thank you for joining us, and thank you to the Gladstone Institutes for virtually hosting us this year. I'm just glad that we can all be together, even though we're apart this year. This is my second year as the Chief Operating Officer at the Golden West chapter of the ALS Association. And during my time, I've experienced firsthand a spirit of community that is truly remarkable. At the Golden West chapter, we're proud to say that everything we do advances the search for effective treatments and cures for ALS. And at the heart of this is our staff. Every day, I see talented, compassionate people throughout the organization working tirelessly to do their part to defeat ALS, all driven by our core value that people with ALS and their families come first. I thank our dedicated team for their compassion, selflessness, and excellence. This year alone, they have provided assistance to more than 1,800 people living with ALS and their families in our service area. In turn, it is the people who are facing this disease who are helping to drive progress in the fight against ALS, and we are celebrating one of those individuals here today. During his nearly decade-long battle with this disease, Jim became an instrumental voice in the ALS community. He helped create the California ALS Advocacy Committee, whose work resulted in the passage of two important bills. The ALS Standard of Care Bill, which stated the highest standard of care for people with ALS in the California Health Code, and the California Tax Checkoff Bill, which to date has raised more than $600,000 for ALS research. In addition, along with others, Jim helped with securing a pivotal $18 million grant for ALS research from the California Institute for Regenerative Medicine. The ALS Association and the Golden West chapter recognized Jim for his work by honoring him with the Iron Horse Award in 2010 and the Heroes Living with ALS Award in 2011 and again in 2016.
Whether it was from his years playing football at the University of California, Santa Barbara, his military service as an Army captain in Vietnam, or his 30 years as an attorney, Jim Barber understood the importance of working together towards a common goal.
Successful teams involve people where everybody on the team puts in more than they take out.
It was no surprise to family and friends that he would turn the news of his ALS diagnosis into an opportunity to help others.
You could see that something had to be done because the progress was not being made, and one way avenue was through the legislature.
My name is Jim Barber. I have ALS. I'm here today with my wife of 43 years, Cheryl, without whom I would do absolutely nothing these days. This disease is a toughie. Research is the only way out. We need some help.
The motion is to pass. The amendments will be taken in appropriation.
Jim worked tirelessly to advocate for ALS research funding beyond the state legislature. He offered key testimony to help secure a pivotal $18 million grant for ALS research.
I feel we're in good shape with this particular proposal to make that happen after 10 years of work.
We all have hope, but hope is not a strategy. Working with people like Dr. Svenson is a strategy.
I've waited a long time to be able to say yes to this.
Jonathan Thomas.
Yes.
Art Torres.
Aye.
Dr. Svenson, congratulations.
Jim and Cheryl participated in many public awareness and community outreach events. In 2015, Jim's family took the ALS Ice Bucket Challenge in his honor. Jim's most visionary and enduring work was the creation of the California ALS Research Network. The active work group hosts the annual California ALS Research Summit. His passionate efforts to advance research, patient care, and public policy initiatives are his lasting legacy that will benefit the ALS community long into the future.
Jim would want you to continue all that you do. ALS awareness, research, patient and caregiver support. A cure was not found in his lifetime, but progress has been made. So please keep up your good work, and thank you.
Jim Barber held a deep passion and commitment to the ALS Association's vision to create a world without ALS. It was his desire that the annual ALS Research Summit would encourage collaboration and sharing of ideas, as well as give special recognition to emerging researchers. Each year, the Barber family and the Golden West chapter present 3 cash awards of $1,000 to each of the top poster presenters. And a couple of days ago, many postdocs and early career scientists presented their ideas in a robust poster presentation session at the annual California ALS Research Summit. 3 individuals were selected for their achievements. Please welcome the winners of the 2021 Barber ALS Research Awards.
Thank you so much, Sherry and Sparrow, for all you do for the ALS Association and introducing this section. And very excited now to go to the poster award ceremony again. This time we're going to do it live for you guys. And I think the idea is then we're going to be going through the poster winners. And there were 3 this year, as there are every year. And we can, let's see, we can bring them up. There were 3 this year. One was Sandra Diaz-Garcia from UC San Diego. And we have Deepti Lal from good old Cedars-Sinai, my neck of the woods, and Shan Liu from UC San Diego. And I think we have everybody here, which is fantastic. So, okay, guys, well, who's going to go first? Did we make a decision, or is Sandra, you're at the top.
Okay.
So maybe we can bring your poster up if someone can bring it up.
Sure.
And then we can chat a little bit, Sandra. I I know your work is very close to my heart because the protein you're working on is involved in ALS and seems to be very exciting for the cells. And this is somewhat a little bit of a collaboration with our group as well. But you know, tell us just in a sort of elevator, no, high level pitch of what you found.
Okay, so what I found is a protein called ELAF3, which it's highly expressed in motor neurons and regulate a lot of RNAs targets. And in ALS neurons, this protein is highly depleted in the nucleus and also in the cytoplasm. So we also find that not only these alterations happen in sporadic ALS, but also in familiar cases. So that we thought that was very interesting, but because not all the familiar cases share the same alteration in some of these proteins as TDP43, which is a hallmark of ALS. Also because this protein can form aggregates within the cells. And
tell me what an aggregate is. Is that good or bad for the cell, do you think, Sandra? If a protein is, and a protein is a structure which every cell has lots of proteins in. What's an aggregate? That looks bad.
So an aggregate is when this protein is not doing its function in the nucleus in this case and goes to the cytoplasm. So somehow, I think that we don't really understand yet how it clamped together, it gets misfolded, so it lost his natural structure, and it start like get kind of sticky and get clamps or even fibrils that could be toxic for the cell.
It's like killing the cell.
Right.
Something got wrong with the protein and killing the cell.
Exactly.
That's quite common. You see that in patient samples.
So that's pretty common. So the most common hallmark is TDP43, but we found that this protein is also very commonly forming these kind of dot-like inclusions in the cytoplasm of the motor neurons, which are the cells that are more affected in ALS.
Okay. We've got like a minute left. You want to go to the conclusions maybe and see what you found?
Okay. So the conclusions that we got from this work was that the nuclear clearance of ELAF3 in motor neurons was a common feature of ALS disease, and its depletion was even more commonly observed than TDP43 pathology. Also that from all the family members of this protein, which share high homology in sequence, ELAF3 nuclear clearance was the most consistent result, and we think that the pathology of this protein was unique in ALS. So hopefully we can understand what is triggering the mislocalization and depletion of this protein and be able to either make this protein back to the nucleus, relocate this protein and restore its function, or understand what is triggering this depletion to generate future treatment for ALS patients.
Right, it's pretty amazing. I mean, you found that this protein should be there. It's kind of a good thing, right?
Right.
Motor neuron, but for some reason it's not there anymore, whether it's to do with TDP43, but sometimes we need proteins in the cell and other times you don't want them. And this one is a good one early on. So if we could figure out how to get more of it, you might be able to change and block things from happening. That's really exciting, Sandra. Exactly. Great work.
Thank you very much.
Congratulations. I can see why everybody was so excited. All right. Next up is whoever, Deepti or Dr. Lal or Dr.
Sure, Clive. I can go next. I'm just going to start sharing my poster. So.
Great. Now I have to disclose here, Deepti was in Bob Balo's lab at Cedars-Sinai, but Bob Balo went off to Roche to head up Roche's all of neuroscience for Roche in Basel, and Deepti has just moved into my lab. So Deepti, I'm going to
correct.
A little high level of what you found.
Yeah, correct. As you know, so my poster basically focuses on microglia, which are the sidekicks to the neurons. You know, neurons die in the ALS, but it's the microglia, which are the resident immune cells of the brain. They actually are involved in neuroprotection. So, and my research really focuses on the gene C9orf72. And as you all know, that leads to, you know, the mutation in that gene leads to all these aggregates and motor neurons, which eventually die. But it also leads to a decreased expression of this protein, the C9 protein in the cells. And what is not very well known in the field is what is this less expression of the C9 protein does to the cell. Is it detrimental? Is it detrimental to the neurons? That we know. But what does it do in microglia, which are actually protecting the neurons? And what I found with my study is that if you have a loss of this protein in the neurons, it actually in microglia, sorry, it actually changes the profile of these microglia. So they are neuroprotective, but when you have a loss of this protein, they're not doing the job properly. They are not doing the protection properly to the neurons. And that is very critical because it's eventually the motor neurons that die, but you don't have any safe basket around them to protect them by these microglia. So what came out of the study was, and eventually it might lead to, all that neuroinflammation that we see in the patients. So the whole idea of this study is that a lot of gene therapy approaches for C9 ALS to cure C9 ALS is trying to target the repeat expansion in this gene. And there was some concerns that whether this gene therapy actually alters the structure of the whole C9 gene, that it leads to reduced protein levels. And what I can say from my studies that we have to be careful in these gene therapy approaches not to really have a more decreased expression of the proteins which are involved in the survival phenotypes for the neurons, because you get other side effects. So, and that's very critical because, yeah.
Just to stop you there, it's really important to get that over to the patients. So C9orf, it's a protein that is made, and it does good things. So in normal circumstances, you need that protein. It's very important for motor neurons. But then it kind of has a problem. It's got these repeats and it doesn't do the right thing. So in ALS, it looks like it's gone off the tracks a little bit and is doing bad stuff.
Correct.
So somehow you've got to get rid of the bad effects without getting rid of the good effects, which is a challenge, right? And I think your data is really showing that if you get rid of all the good effects, and then you've got the motor neuron, but you've got the support cells for the motor neuron, the microglia, which are looking after them. And if you knock it out, if you affect the microglia, you might do something bad when you think you're trying to do something good. This is why science is complicated, everyone.
Yeah, and Clive, you're correct. It's mutation is present in all cells, right? Neurons and microglia. So it's affecting all the cellular functions. So we have to be really, investigate all these functions in different cell types and to see how all these cell types contribute so that we can have effective therapies to actually cure the disease. And not focusing on neurons is very important, but other cell types also play a very critical role for all that neuroprotection.
I've got to rush to another poster, Diti, but do you want to give me your...
Sorry.
And then we'll move on to Dr. Liu.
Yeah, so what I found, and this is actually the summary, is that you have altered microglial function due to loss of C9 in microglia that actually can contribute to neurodegeneration itself by causing the synaptic defects, which I talked about, and also it can cause toxicity, which is independent of all that aggregation that is happening in the neurons.
So we have to be careful of gene therapies, because this is why safety is so important, and these studies are so important. That your work is showing us that we have to think carefully about just putting a gene therapy in, because it could do something bad that we didn't expect.
Correct.
Now, based on your data, we can target it a little bit differently to avoid this problem and still be successful in treating the patient. Wow. Correct. That's why you got the award. I love it.
Thank you.
An amazing story. Okay.
I would like to thank the ALS Association Clive and the Jim Barber family for this honor. Thank you very much. It motivates me even more to work on this disease and find a cure.
Thanks, Steve D, and you're fantastic, and it's both of you fantastic work. Now, at this point, I normally go to the bar and have a gin and tonic, because normally poster is in the evening. So by the third poster, I'm really getting going and having a lot of fun. So I'll just imagine that I'm having a nice evening. And now we get to the last poster, because these are usually in the evening, guys. This is after the meeting. So Dr. Liu, tell us about your poster.
Everyone, so I'm a postdoc fellow at the Cleveland Lab at UCSD. So we are working on the mechanism of TDP-43 pathological structure formation, because we all know TDP-43 is one of the most common pathological hallmarks of ALS disease. So it happens in almost all the ALS patients. So to understand the mechanism that how this pathological structure formation is important for finding treatments for this disease. So in our lab, we generated like cellular models to that can mimic the disease, the TDP-43 aggregate formation, so in the cells. So we found that this stress can induce TDP-43 from those TDP-43 demixing structures in the cytoplasm, and independent of structures that called stress granules. And we also found this structure
Dr.
that
Liu, how do you, so stress, I know I'm stressed all the time. How do you stress a cell?
So here we use a stress that has been used in the field that to induce stress granule formation. It's called sodium arsenide. So if you know arsenide is kind of a toxin to everybody. So, but yeah, this is like a common stress.
Like arsenic, you give the cell poison and now it's stressed. Yeah. And then when you give it the poison, this TDP suddenly goes up and it's like responding to the stress. Just like in a patient, maybe, we don't know what the stress is, but TDP also is going up in ALS. Okay, I get it.
Yeah, so and also in the stress condition, it forms those aggregated inclusion structures. So and also we found that so in this in my study we're focusing on finding a mechanism that regulates this process. So we're using like approach to identify the binding partners of TDP-43, and we found a protein that's called HSPP1 that binds to TDP-43 in those demixing structures, and we also interestingly, this HSPP1 protein is highly expressed in motor neurons, and it's also been found to be mutated in myelin motor neuron disease. And also, this protein has also been found in several ALS sporadic ALS cases. So suggesting HSPP1 plays an important role in the function of motor neuron. So collaborating with Dr. John Ravi's group, we also want to see what happens in the ALS patients with this protein, and we found in the motor neurons with this TDP-43 pathology, there's a decrease of this protein HSPP1, both in two of the ALS cases. So next we want to see if knockdown decrease of HSPP1, whether it can promote or induce the cytoplasmic TDP-43 demixing. So we found in the cell model that we generated, we found knockdown of this HSPP1 could increase the TDP-43 cytoplasmic demixing structure formation. And also we found it can also cause TDP-43 mislocalized to the cytoplasm. So in conclude, we found this smart HL protein binds to cytoplasmic TDP-43, and motor neurons also express high levels of this protein, and this protein is important for motor neuron function, and decrease of the protein also happens in the motor neurons with TDP-43 pathology. So in conclude, we think this protein may be a potential therapeutic target that can mitigate TDP-43 pathology in ALS patients, and it can be helpful to the disease like therapy development.
I like it. I mean, I think the nice thing here is with collaboration as well, is when this is what we do in the field. This is why this meeting is so important, right, Shan? Because you had a question, you saw this protein changing, and then you went to John Ravitz, who's a pathologist, and said, do you have any brain samples from patients I can look at to see if it's, and we do that all the time. And it's really important for everybody to understand how the ALS Association, through funding all these different research projects and getting us all together, means we can now run away from this meeting and go, oh, I'm going to collaborate with this group. And you three scientists are probably going to collaborate more now. You've heard each other's data as well, because we're all working as a team. And I think this heat shock story is great. I mean, can you imagine this going into a drug? My last question, and we'll finish up, but how would this maybe be made into a drug of some sort? If I gave you $1 million tomorrow to make a drug, what would you do?
Yeah, I think in the field now there's also some drugs that increase the HR protein chaperones that had been in clinical trials with ALS patients. So I mean, for this interesting thing is like those drugs, they do not increase the levels of this small HR protein family. So we can also combine maybe like AAV mediated like gene therapy that both increase those other HR protein chaperone system, combining with this small HR protein that can maybe boost the effect.
Very exciting. I think the other exciting thing is that this discovery, and we heard from Dr. Okano from Japan, that if we could make stem cells from each patient and find out, because maybe only some patients have this heat shock protein issue, and other patients have C9 or like Deep told us, and other patients have ELAVs like Sandra. So maybe there's 3 groups of patients. If we knew who they were, we could develop drugs for those patients, right? And I think iPS technology, the stem cells, making stem cells from each patient could make us understand better which drugs should be applied to which patient. We had a whole discussion about that yesterday, which was fantastic with the clinicians, how they would love to have more information on the patients before they just get another 100 patients into a trial, whereas each patient had a different type of disease. And ALS may be clustered into these groups. And I think your work, all three posters are really taking us closer to understanding more about the specific types of disease. It looks very cloudy now, guys, right? But when you take this from a high level and start looking at all the data, you start seeing these subgroups of ALS coming out through the kind of work you three are doing. Hats off, fantastic work. And the Barbara Award would be proud for you guys to get this, and thanks to the ALS Association. All right, we'll close this session out. Thanks very much. I would be a round of applause if we could hear all the clapping. It would be going crazy right now. And I'll hand back to Audra for the last questions. I know we're a little over, guys. I'm sorry, but that was a lot of fun.
That's okay. It's worth being over for a few minutes. Big congratulations to all the Barbara Award winners. Thank you guys so much. Those were wonderful presentations and a little over my head, I know, but amazing information. So thank you so much. One or two more questions, Dr. Svenson. One person said that you mentioned the gut biome research. Just wondering if you could talk a bit more about that. Related to that, someone else is asking, is there viral or bacteria introduced through the gut, through the intestines, that might be causing inflammation, could be causing ALS?
So there's a study being done at Mass General where so the gut has been very involved in Parkinson's disease. They've really got a strong association. In Parkinson's, you actually get constipation and gut problems almost 30 years before you get Parkinson's. So we know there's an association in Parkinson's. We're just learning about ALS. It doesn't seem from the data that there's a strong effect of the gut in ALS, but there may be a modulatory effect. What I mean by that is if you're starting to get ALS and your gut is in a certain condition, like you have a lot of microbiome. The microbiome is these bacteria that live in the gut. If you've got a whole load of bad bacteria and you're starting to get ALS, there could be an interaction through those two things. And we're trying, and there's a few studies now, and Kevin Egan from Harvard presented some data that in mice, there's a mouse model of ALS where the mouse gets ALS. Well, it would always get ALS if it was at Harvard. And they moved the mouse to MIT and it didn't get ALS anymore. Well, that's crazy. We put a genetic deficit in it. And it turned out that the Harvard facility was much dirtier than the MIT one. MIT folks are very clean and crystal clean. The Harvard guys, a little not so much. It's an old, like Cambridge university. Yeah. So their animal facility had all these microbes in it, and MIT spanking new didn't. So it turned out those microbes were interacting and causing ALS in the mouse, and it could happen in humans. So we're trying to understand that association more.
Wow, that's very interesting. I did not know that about MIT and Harvard. So that's
don't let it out. It's very confidential information.
Thank you. One last one. Is there any new information or eligible studies for patients with really slow progression ALS? He's defining that as 16 years since onset and not depending on any aids such as wheelchairs or walkers.
You know, that's a great question. And what's happening in all the clinical trials we heard yesterday and the day before is where they get responders, it tends to be the rapid progressors that respond in these trials. Now, of course, these trials are usually 6 months. So if you're declining very rapidly, you're more likely to see a change when you're declining rapidly. But if you're going very slowly, you might not be able to see a difference in just 6 months. I think we need to do longer trials and we need to go into patients that are later in the disease. They often get excluded if the inclusion criteria are often you must have had ALS within 2 years, or I feel like we need to do a lot more work in those patients. I do think the kind of things we're doing, the transplanting stem cells into the spinal cord, if we get it working and it's safe, if we want to give a boost to a patient that's clearly stable, one can imagine doing that to see if the GDNF or the drug might help them. And we need to develop more trials for these longer haul patients with ALS. I agree. They tend to get left out of the new trials. We need to address that. And maybe next year that could be a topic for one of our sessions. So I thank whoever asked that question. You might have stimulated a whole set of new discussions around long-term patients and trials, and we need to address your needs as well.
That's great. I love that. Well, thank you so much, Dr. Svenson. You took a lot of time with us this morning, and thank you again to the 3 award winners. Thank you guys so much for your time presenting your wonderful posters, and feel free to stick around if you'd like to for the rest of our program. But now I'm going to introduce Dr. Steve Finkbeiner to join us for another presentation. Dr. Finkbeiner is the director of the Center for Systems and Therapeutics and a senior investigator at Gladstone Institutes. He's a professor of neurology and physiology at UC San Francisco, and his expertise encompasses neurodegenerative and psychiatric diseases, iPS cell modeling, human genetics, robotics, imaging, computational methods, and artificial intelligence. Please take it away, Dr. Finkbeiner. Thanks for being here.
Great. Thank you so much, Audra. Can you hear me?
Yep, can hear you just fine.
Terrific. And you can see the slides?
Yes.
Excellent. Well, it's such a pleasure to be here with everyone. Thank you so much for coming this morning. It was my pleasure being the host this year for our California ALS Research Summit. You heard a beautiful kind of review from Clive, and it was another exciting meeting. I'm a neurologist and a neuroscientist, and I think one of the things that I love so much about this meeting is it's one of the very few that really try to bring clinicians and basic scientists together. And I think what's really rewarding is that both sides really try to reach out and communicate with the other, and I think help form a team that will be very effective for trying to tackle ALS. I only have a few minutes with you this morning, and I did not want to go into a deep dive into a lot of the nitty-gritty of the research. You got some beautiful poster presentations there a few minutes ago. It gives you a flavor of kind of science and sort of trying to tackle ALS at a molecular level. I thought maybe instead this morning what I would try to do is just cherry pick a few highlights from our research summit. And I thought a theme that kind of emerged for me that I thought the patient community and families might be interested in are talks that touched on things that were really relevant for overcoming obstacles to improve the diagnosis and treatment of ALS. So one of the things that you may have heard about are these things called platform clinical trials. And this has really been an exciting development. It's begun to appear in other fields, but now really getting established in ALS. And it really helps to, I think, improve the number of things that we can test and try for ALS patients and hopefully lead to the development of therapies that are going to be effective more quickly. So with conventional trials, generally one drug or a combination of drugs, you heard a little bit about this from Clive, gets tested one at a time. And that's very slow. And in fact, it's slow partly because for each trial, the whole structure to do the trial has to be built up from almost scratch for that trial. So enrolling the patients, hiring all the staff, et cetera. It's almost like if you were going on vacation and you had to build a house every time you wanted to stay someplace for a night. And what platform trials do is they really change the whole structure. It's more like having a hotel that's already built that you can go visit and leave whenever you want. So there's already a huge infrastructure set up to be able to test agents. And there are clever ways that clinical scientists have been able to structure these trials to enable more things to be tried at once. You heard a little bit earlier about TUDCA and this idea of testing combination therapies. It's easier to do in this structure. And it also means that in terms of placebo groups, we can be able to share information that we collect sometimes from single placebo groups that can then be used across multiple trials, and that enables us to compare more things. So I'm very excited about this. I think this is going to be a major development for ALS. It'll make things less expensive to do clinical trials, and that's going to mean that we'll be able to test more things. And I think that naturally will lead hopefully to more effective therapies, but also will teach us a lot about what we need to do to be able to really effectively treat ALS. Now you heard this morning a couple people talk about really trying to understand the structure of ALS, the disease. You have to keep in mind that we neurologists tend to make these diagnoses based on patient symptoms mostly. And so one of the open questions for ALS is, we know folks who have ALS all have certain symptoms in common, but do they have the same underlying cause of ALS in common? In other words, is ALS one disease, or is it many subgroups of diseases that all have a common sort of clinical syndrome? And the answer to that question is really important, because the way we do clinical trials now assumes that every patient who's in a clinical trial stands to benefit from the therapy. But if there's a therapy that only works on one mechanism of ALS that's only relevant for a subgroup of patients, then the way we do clinical trials will have a very difficult time detecting it. We may see a few folks get better, but in the context of a larger group that doesn't benefit, statistically, we can't prove that those few who benefited actually benefited from the therapy itself. And so unfortunately, in that scenario, we could end up with a drug that actually did work, but we were unable to show it. It wouldn't get approved. And in fact, the scientific community would tend to regard the target of that drug as maybe irrelevant. So trying to figure out if there are these subgroups of patients within ALS who may have a common cell function problem that might merit a particular therapy is really important. But it turns out trying to figure out this question is very difficult, because unless you already know the mechanism that these patients share, it's often hard to figure out or identify subgroups of patients that share them. And I wanted to just say briefly about a very big project that Clive and I are both involved with called Answer ALS, where we're really trying to do this. This is the main focus of the project. For those who haven't heard about this project, this is a multi-center project led by Clive and Jeff Rothstein, but involving researchers all across the United States, where we've enrolled over 1,000 patients, taken really deep clinical histories and followed these patients over time, doing exams and characterizing them. And then we have made many measurements from these patients. My group has particularly been involved with looking at their DNA sequences and genomics and genetics. We have also used monitoring devices. And then Clive's made IPS lines from each of these patients. And we're using these IPS lines to make motor neurons that we can then study with a variety of other techniques to get additional data, including work that we do here at Gladstone studying imaging. And what we think is that we can, together with this very large data set, begin to look within that large community of patients to see if we can identify signatures that point to ALS. And then if we can dive even more deeply and find subgroups of patients that may give us clues about a common mechanism and that we can use to develop therapies. And at this meeting, someone from my group already, I think, presented some really exciting new data where we've begun to study the genetics of patients in this large community and have already begun to identify subtle variants. So not actual gene mutations. And you've heard about a few of those like SOD1 or C9orf72 that can cause ALS, but still are relatively rare and may affect fewer than 10% of patients with ALS. What we've done instead is to really look at the genetic sequence of patients who have ALS but don't have any of those known gene mutations. And yet we've still been able to discover genetic variation in their DNA that can predict risk of ALS and even maybe the rate of disease progression. And so we're very hopeful that continued work on this will continue to yield new insights and fruit about ALS. And if you want to learn more, there's a website here, answerals.org, where you can learn a little bit more about the effort. I saw several earlier questions about CRISPR and other genetic strategies, and it was my great pleasure to invite Jennifer Doudna. As many of you know, she is a professor at Berkeley, but a few years ago, she moved a big part of her lab to Gladstone, and she's a couple floors above my laboratory, and we collaborate. And what Jennifer did was to discover an innate immune system in bacteria to fight viruses called CRISPR that she and others have cleverly adapted to be able to essentially create sort of surgical scalpels to go into the genome and be able to correct or change specific DNA sequences. And as you heard, she won the 2020 Nobel Prize in Chemistry for this, together with Emmanuelle Charpentier, the first two women, frankly, who've won the prize. And so that was quite an achievement. And we're excited because, as I just said in my last slide, it's true that about 10 to 13% of ALS patients do have mutations in specific genes that cause ALS. But we're also aware that the fraction of heritability of ALS, in other words, the chance that you'll get ALS is elevated for patients even without those mutations if they have a family history of ALS. So we know that there are genetic roots to ALS, and I think this CRISPR technology adds an exciting, new, very precise way to manipulate the genome to some of the other tools that you've already heard about that we are bringing to bear with ALS to be able to regulate genetics. You've heard about the antisense oligonucleotides, some of which are in clinical trials, as well as viral delivery of agents from companies like Avexis, now Novartis. And so I'm very excited about that because I think as we understand the genetic underpinnings of ALS better, we're going to have a whole toolbox we can use to develop therapies that very precisely target those mechanisms. And I think that gives us great hope that we not only will have something that is on target, but something that we hope will prove to be effective. Then I just want to finish my brief talk talking a little bit about an area that's exciting to me, and that is harnessing artificial intelligence to help patients now. And I think we're very fortunate to be in California, and especially the Bay Area, because we have so many amazing information technology companies around us. And my laboratory was, as far as I know, the first academic collaborator with Google. We started a number of years ago collaborating around a number of projects, but actually especially focused on ALS. And so that was a theme in this meeting, how AI and some really powerful new computer biology and science can help us tackle ALS. One of the nice things about this meeting, in addition to bringing clinicians and scientists together, is that we also often take the opportunity to bring people in from outside the ALS field who may have exciting insights or new ideas or approaches from other fields that we could bring to bear on ALS. And so we had an expert from the field of oncology or cancer who's really been useful for developing personalized medicine approaches to patient stratification. Some of you may be aware, for example, that for many years we thought breast cancer was just one disease, but then as oncologists began to investigate, they discovered that different breast cancers had different markers, and depending on which marker, you may be eligible for a different therapy, and the efficacy of the therapy varied tremendously depending on getting the right marker and the right cancer. And so in many ways, the cancer field is ahead of the ALS field in this regard. And so we asked an expert to talk to us about how to use some of those insights and approaches. And then I wanted to tell you a little bit about another project that Google has developed a couple of years ago and that you can access if you want, called Euphonia. A little video here I'll just play to help use AI to help develop assistive technology for patients that can be used today. But I think a lot of these tools also are going to turn out to be very helpful for clinical trials in the future.
Google has very good general speech recognition, but if you do not sound as most of people, it will not understand you.
No one's ever collected large data sets of people whose speech is hard for others to understand.
People who have multiple sclerosis, for GF, who had stroke, who stutter.
They're not used in training the speech recognition models. The game is to record things and then have it recognize things that you say that aren't in the training set. Dimitri recorded 15,000 phrases. It wasn't obvious that this was going to work. He just sat there and he kept recording.
We need to make all voice interactive devices be able to understand any person who speaks to them.
you can see that it's possible to make a speech recognizer to work for Dimitri. It should be possible to make it work for many people, even people who can't speak because they've lost the ability to speak. The work that Shenxing has done on, voice utterances, from sounds alone, you can communicate. But there might be other ways of communicating.
Most people with ALS end up using an on-screen keyboard and having to type each individual letter with their eyes. For me, communicating is slow.
Steve might crack a joke, and it's related to something that happened, a few minutes ago. The idea is to create a tool so that Steve can train machine learning models himself to understand his facial expressions. To be able to laugh, to be able to cheer, to be able to boo. Things that seem maybe superfluous, but actually are so core to being human.
I still think this is only the tip of the iceberg. We're not even scratching the surface yet of what is possible. If we can get speech recognizers to work with small numbers of people who learn lessons, which we can then combine to build something that really works for everyone.
To understand and be understood is absolutely unbelievable.
Great, and I think I'll end there. Thank you guys so much for your attention. Something, folks who are in the Bay Area or can make it are always welcome to visit the Gladstone. We're happy to give tours. Maybe not now with the pandemic, but maybe when the virus gets better. We love to do that. And a part of it for us is that we deeply believe this really is a team effort. You've heard today about work that researchers do, and Clive painted a pretty sad story of penniless postdocs doing the work, which is sadly true, and especially in the Bay Area. But I want you to know that everyone has a role to play. The patient community is absolutely essential, and groups like the ALS Association and philanthropists are really critical. And then together with academia and industry, I think the team is really going to be the way we're successful beating this. So thank you again.
Thank you, Dr. Finkbeiner. We have a couple of questions. Dr. Svenson, I saw you pop back on. Did you have any comments?
No, I'm just listening. Go ahead.
Okay. All right. Wonderful, wonderful. A couple of things that came in, Dr. Finkbeiner. I know ALS and Parkinson's was touched on just a little bit earlier, but we have a couple of questions around that. Just one woman is saying, my dad has ALS and his mom had Parkinson's. Is there a connection that we know of yet?
Yeah, that's an interesting question. So it certainly is the case that it's been our feeling that there are common threads that cut across multiple neurodegenerative diseases. There are a number of genes that are risk factors, for example, for a couple different diseases. And for ALS has a very close relationship with another neurodegenerative disease called frontotemporal dementia that many people may have heard of. We understand some of the genetics, I think, of FTD, and one of the underlying themes or common threads that does seem to cut across multiple neurodegenerative diseases is this issue of being able to handle proteins that misfold and clump. You heard a little bit about that in the poster presentation. And there's a pathway called autophagy, which seems to be especially important for that. Another common thread is neuroinflammation, and you heard about that. I see a couple of questions in the chat box from folks who were asking about that, and that has also emerged as a common thread between Parkinson's, for example, and ALS.
Great, that's really helpful, thank you. Kind of a broader question, has anyone seen success from stem cell therapy?
Actually, if Clive's on, he may be able to address this better than I can. There are a number of stem cell trials in other disorders that have reported beneficial effects. Clive, do you want to comment?
I'm happy to comment. And the unfortunate answer is no. I mean, in simple terms, there isn't a stem cell therapy yet on the market that works for ALS. I think the Neurone trial, the Brainstorm was the last one really that reached phase 3. And like I mentioned, it unfortunately didn't reach its endpoint. And so that's not going to be advanced right away. But there were some signs in that stem cell trial that when they put the stem cells into the brain, the fluid in the center of the brain, there were changes going on which suggested they were doing something good. Unfortunately, that didn't translate, Steve, into a clinical outcome that was above a point at which the FDA would say, "Oh, go give that as a therapy." Didn't quite reach it. So we need to do more work. I think that's the answer. But there's lots of interesting trials going on, but not there yet.
Yeah, and just to add to Clive's comment, Clive's absolutely right. We don't have anything to show for ALS yet, but there have been some efforts in eye diseases and elsewhere where there have been some promising findings. So like Clive, I think we're hopeful, but more work needs to be done.
And just to say, the investment that the ALS Association has made in this space, including me early on, has been enormous. And I think, unfortunately, so you think about, you're using a cell as a drug, essentially, and it's going to try and do all the stuff it normally does. It's like, talk about combined therapy. If you put a new cell in the brain, it's making all the stuff that it should. So, but that also is very complicated, and getting the cells really to the right place to do that is what's been difficult. And that's the same for CRISPR, is getting the editing to every single cell is one of the challenges that we have, basically, Steve. And so that's an interesting area.
Wonderful. Thank you both. Dr. Finkbeiner, I wanted to thank you too for showing that video on Project Euphonia. Our Golden West chapter has been marketing that program, and we've sent several folks who have participated in that program. So it's really helpful just to see that overall. It was a great video.
Yeah, thanks, Audra. And just if I can add to that too, thank you. Just encourage folks to participate if they can, because the power of artificial intelligence is really driven by participation. So the more people who participate, the better it gets. And you saw an example there where Google was trying to use AI initially to just help understand speech that may be impaired. But what they're trying to do ultimately is to be able to create new ways for patients to communicate with the world, even when they lose their speech altogether. You got a little sense of that at the end with the way it's possible to communicate even with some facial expressions. And then in the long run, what I didn't really get to tell you in that slide, but that came up during the meeting, is they're trying to develop whole new ways to be able to use the phone and other devices to be able to measure many things about patients' movements that will be, we think, very useful for doing clinical trials. So those of you who've participated in clinical trials probably know what it's like. You go for a short visit, someone does an exam that's fairly brief. We know that that's very limited. Patients have good days and bad days, and sometimes it's really hard to get a good measure of where a patient's at. So we think these more comprehensive ways that allow us to monitor patients over time are going to give us measures that are going to be much better for doing clinical trials. And so we also think, together with that platform clinical trial approach, we may have a lot of new tools that will reveal, hopefully, more effective medicines.
Mm-hmm. Great. I have one last question for you, Dr. Finkbeiner, a little bit of a different topic, but is there a desire by ALS research scientists for donation of body parts and organs?
Yeah, so that's a great question. In fact, that is a really important part of our research because, as Clive probably mentioned, and as you got a little sense from the poster presentations, a lot of the work we do in the lab necessarily has to focus on cells in a dish or relatively simple models, even mouse models. But a really critical piece is connecting it back to patients and making sure that whatever we discover in a dish or in the lab really is relevant to ALS. And the only way to do that is to be able to ask that question and investigate with the help of ALS patients, including tissue from ALS patients. So that is something that's very important. And we have a number of facilities on the East Coast and on the West Coast for being able to bank material that we receive from patients and then distribute it to researchers so that we can do that important work.
Wonderful. I know we have a couple of questions just about how to participate in Project Euphonia. We'll put the link to that site into the chat so that people have that. So
terrific. Thank you.
Thank you for being here, Dr. Finkbeiner. This was a wonderful talk. You encapsulated so much of that summit so well in a brief period. Thanks for being here. Much appreciation to you around that.
My pleasure. Thank you. Thanks, everyone.
Yeah, absolutely. I am pleased to introduce Dr. Jeffrey Rosenfeld for our last section of our program today. Dr. Rosenfeld is the professor and associate chairman of the Department of Neurology and the director for the Center of Restorative Neurology, Loma Linda. And he's director for neuromuscular ALS programs at Loma Linda University Health Systems. He created and developed the Center for Restorative Neurology at Loma Linda, which involved organization of a large multidisciplinary team for full-time comprehensive care of patients with a variety of neurological disabilities. So thank you for being here, Dr. Rosenfeld. Take it away.
Oh, there we go. Well, thank you very much, Audra, and thank you all for being here and for showing an interest and learning about what we've done over the last couple of days and what's going on in the field. I've been very fortunate to be part of this conference since its inception. It's one of the advantages of being older in the field, I guess, but I think I've been to every one of these since they started. And it's really a great conference, as you've heard about, for many of the reasons that have already been brought up. It's a great way for those of us who spend more time in the clinic or less time in the clinic to associate with our colleagues. And then usually when we're in person, the patients are just down the hall in an adjacent room, so you get that perspective as well by coming in to meet many of you. It's gratifying to me to see so many of the patients that I already know on the list of attendees as I scrolled through earlier. So hello to those of you who I know, and welcome to others. My part of this conference in the last couple of days has been a little different than the rest of the conference. I organized a session that was a critical evaluation of where we are with ALS clinical trials. Perhaps it's one of those advantages, again, of being around for a little while, that you can kind of step back and say, are we going in the right direction? But it was a little different than than many of the other talks, because it's a very upbeat conference, lots of progress, lots of development, and everybody's excited. And this talk was about, let's just hold on a minute. Are we going in the right direction? And if we're not, where should we be redirecting? So it was designed as a debate. I had 3 colleagues with me from industry as well as academia, and I had assigned them, this was so funny, I had assigned them a position to take on each of 5 different themes that I'll show you in a moment. And asked them to argue the theme that I gave them, even if it wasn't their personal opinion. And academics, as they are, didn't always stick to the script. So it made for an interesting discussion, but I'm hoping in the next couple of minutes to highlight for you some of the things we think about when we're trying to decide if our efforts in clinical trials are in the right direction or not. And then, of course, I'll spend all the time needed to answer the questions that you have. So this slide is a list of compounds that have been tested in ALS. It's not totally up to date at all. In fact, this came from a publication about 6 years ago. So if it were totally up to date, I'm afraid the second column would reach down more towards the bottom of the screen, because we've tested a lot of things. I've been in the field for over 25 years, I think, and I've been involved in a lot of trials. And I'll tell you, of course, at different stages of your career, you have different feelings, but many of these drugs went into clinical trials with great enthusiasm. And that enthusiasm generally came from basic science work that we thought, gee, that worked so well in that model, or that worked so well in a few patients in a earlier phase trial, that it's ready. Boy, we're on it. We're going to definitely make a difference, only to find out, as many of you well know, that they didn't work. And the only ones that did show some efficacy to work are riluzole, which was tested and approved in the 90s, and edaravone, Radicava, which is approved just a few years ago. But in between riluzole and Radicava, and before riluzole, there have been a lot of trials tested. And so the obvious way to approach this, the way that the knee-jerk, so to speak, the knee-jerk reaction is, we didn't find the right drug. The drug we were testing just wasn't beneficial enough. And honestly, that's been the thought for many, many years. Let's go back to the drawing board. Let's take the next one. And this has been a really exciting field to be in because there are so many ideas. So there have always been so many compounds in the pipeline, including today, when we have perhaps even more than ever compounds that are in the pipeline. So it could have gone without saying that we're on it, but we just haven't got there yet. And it is a difficult problem. The purpose of this portion of the symposium was to say, what if it's not the problem with the drug? What if the drug itself might have worked, but as Steve Finkbeiner mentioned earlier, what if it only worked in a subset of patients? What if we missed it because of the way we designed the test, not that the drug itself wasn't working? And of course, nobody knows the answer to that, but we were trying to evaluate that question. And you know, it goes without saying that, in fact, there's one memorable example earlier in my career. There was a drug long ago manufactured by the Sanofi pharmaceutical company. They did a huge international trial with this drug. It was one of those ones that we really thought we were going to get to the answer. And come to find out that the drug was not effective at the end of the trial. But in years to come, the company allowed patients that thought they were benefiting from it to stay on it for quite some time. And many of us would get together at these regional and annual meetings and share, how many patients do you have that are doing well on that drug, or you have? And you know, there was a subset that really seemed to benefit, as there is in many of these other clinical trials. Now, was that a function of the disease? Perhaps their disease wasn't as aggressive and they would have benefited anyway, or were they a subset responder? So if we go back to some of the earliest trials, this is an article that preceded the approval of riluzole. And it was an article about a trial which showed benefit. And that led to the trial, which ultimately gave the approval to riluzole. And the inclusion criteria for these trials were pretty simple. The age range, you just had to have probable or definite disease. You couldn't have been affected for more than 5 years. You had to have a breathing capacity of 60%. And then they paid attention to whether or not your disease was affecting your limbs or your disease was affecting the bulbar function of speech, swallowing, and sometimes breathing. And that was it. And in fact, they found a difference overall when they did the subsequent study, that it wasn't a dramatic difference, but it was a statistically significant difference, and it led to the approval. Over the years, many, many trials have been done since that time. And what I noticed, and others, I shouldn't say this is a totally novel thought, but what I noticed is we are very consistent in the way we do clinical trials. We generally enroll patients with a similar paradigm to even the early trials in the 80s and the 90s, much like the ones that you just saw, the criteria you just saw with riluzole. We also evaluate patients in a very similar way. We use the ALS-FRS to evaluate patients. We use quantitative muscle testing. We use vital capacity, and we use those as markers of how well the disease or how aggressive the disease is progressing. But what if that wasn't a good marker? What if the enrollment criteria enabled us to miss or resulted in us missing treatment effects? So just to look as an example of what I've been talking about, these are just snapshots of inclusion criteria from recently conducted trials. Some of them are still ongoing. And in this particular one, if you look at this Biogen trial that some of you may actually be involved in, laboratories supported probable or definite ALS. That means we don't have to be 100% certain, we just have to be pretty certain that you have ALS. This trial, the Mallinckrodt's Actar gel, this was a negative trial of several years ago. They tried to randomize the patients into groups, but the drug itself wasn't very effective. And as a result, the ALS-FRS score that they were using didn't show a benefit. This is a ravulizumab trial that's currently ongoing, sponsored by the Alexion pharma company. And here they accepted lots of variability, possible ALS, lab supported, probable, or definite ALS. And then, as you mentioned, as was mentioned earlier a couple of times, the platform trial, which is currently ongoing, is also wide open with clinical clinical certainty. So what's the upside? Well, the upside is a lot of people can get into these trials because they're so open. You can have possible ALS, which means that clinically we see definitely an indication that ALS is a good possibility, but we're not certain. It turns out most of the time we're right, but we're right in that ALS has a great variability of expression, and not everybody has sort of the same type of ALS. But all of those patients are in the same trial being measured on the same platform. It's a very innovative trial because it's going to get us to answers faster, but it's also potentially an unsettling trial because what if we missed an effect that was there because the inclusion criteria were so open? So the session that this was focused on was centered around 5 different themes. And my purpose here is just to kind of give you a snapshot of what the discussion was like on these 5 themes. Well, cohort enrichment means that we're not going to enroll everyone. We're only going to enroll patients that, number one, we think are going to benefit from this drug or that are in a select part of their disease progression. Now, the upside to this is you don't have a very variable treatment group, in that the patients that are enrolled are all in a similar stage of their disease, or perhaps they have a similar type of disease, or perhaps they have a particular biomarker that was relevant to the drug that we're testing. So one thought is that increases your chance of finding a difference. There was a little controversy about this. On the one hand, everybody seemed to agree that enriching the cohort when you knew how to enrich it was a good thing to do. That was logical. But without knowing, that is, without knowing what the best way to enrich the enrollment group is, we might as well take everyone. So there was a little bit of a debate about whether or not we should focus the enrollment groups or not. And from the patient's point of view, I spend most of my week talking to patients. I will tell you that this is a very considerable concern, in that if somebody wants to get into a trial, they don't want to know that they don't have the right kind of ALS to test the drug. They just want to get in and see if it works for them. And I totally understand that. But we have to recognize that that might come at the expense of the sensitivity of finding a difference if it exists. Similarly, another theme was that very early affected patients, that is, patients that are just in the very beginnings of the disease, may or may not be the best patients to test. On the upside, earlier affected patients have more motor neurons that are alive. And so if you have a drug that protects motor cells, that would be the patient you want to focus on, those that have the most available motor cells to be protected. On the flip side, those also are patients that have a lot of variability. Some are progressively, some are progressing fast, some are progressing slow, some might not develop ALS for some time or only develop it in certain areas of the body. So we introduce a lot of variability when we study early affected patients. All of us want to treat every patient. We want to treat the earliest affected patient, but who we want to treat and who we want to test in a clinical trial may or may not be the same person. And that's a very difficult spot that we're in right now. So that led to some interesting discussion along the lines of what I just said for theme number 2. We also spent some time talking about this ALS-FRS function rating scale, because it has been tried and true. If nothing else in this field, we have been incredibly loyal to the ALS-FRS score. And it is a very good score to measure how a person is functionally from one time to the next, with much less subjective than just saying I'm doing better or I'm doing worse. It's actually a number. And the number is fairly linear for a good portion of the disease. And we can kind of predict in a certain person what the rate of progression in their disease would be based upon the rate of decline in their functional rating score. So it's really served a great purpose. But what if it was too coarse of a measurement? What if the ALS-FRS score didn't really measure the benefit of a drug? What if looking at a person's function was too removed from the actual site of the drug action? Well, in that case, we would be in trouble because we really rely on the ALS-FRS. And at the moment, we have lots of discussion in the field about something called biomarkers These are generally biochemical measures that we can take from either blood or spinal fluid or sometimes urine, which are indirect measures of potentially a drug's action. And should we always or not include biomarkers with every one of our trials to see if we have another readout other than the functional rating score? Many of the trials, including several that are going on right now, don't use that. They'll use the functional rating score as a sign of efficacy. And mixed into this concern is the FDA, because the FDA is generally very prudent, but kind of conservative about what they will accept as proof that a drug works. And the ALS FRS is something that's on their list that they would accept. So consequently, pharmaceutical companies like the ALS FRS, as do many of the investigators, because it's already been proven and validated many times, and it actually was involved in the approval of drugs to date. The fourth theme we talked about was the platform trial that we've mentioned before, because the platform trial is very innovative and it certainly will get us answers faster. We will know more about more drugs, and the efficiency of the trial comes largely in that the placebo group is shared between the other treatment groups because everybody is being tested on the same protocol. It's the same protocol no matter what drug is being put into the platform. And it's a dynamic trial because they're always adding drugs. So if you enter the platform trial today, the drugs that you might be potentially exposed to or randomized to will be different than if you enter the platform trial in, say, a year from now. The drugs we're testing would be different, but the ones that we initiated today would still be ongoing in the same platform trial. So it's a great way for us to compare and contrast the way that drugs respond to each other. It's a wonderfully efficient way to get an answer faster, but again, we don't pay much mind to the stratification or the individuality of certain patient types. So this generated a lot of discussion because there's so much enthusiasm, appropriate enthusiasm for the platform trial. And the contrary opinion largely was, we don't have any biological information that's at this point that distinguishes one, so to speak, form of ALS from another form of ALS. And so until we do, we shouldn't try to separate out the treatment groups and only enroll selected groups in a trial like the platform trial. And that point is well taken, although it doesn't change the concern that one has that we might be missing treatment effects. I know that individuality of ALS, the so to speak personalized medicine approach of ALS, has been a very strong interest of mine for many years. It was that interest that generated a symposium that we put on, I don't know, almost 20 years ago now, on combination therapies. And would the only logical therapies for ALS, motor neuron disease, be combinations of drugs? Because each drug might potentially target a different mechanism. And at any point in time, a patient would be affected by one of the other mechanisms in the combination, so the combination would capture more of a treatment effect. It's still a good idea. It wasn't very well received at the original conference because we didn't have as many ideas. It's much more of a common discussion now. In fact, I think you heard it earlier this morning, the notion of combination therapies. And perhaps that's our way of treating this population of heterogeneous, this population of patients that are so heterogeneous in the future, because we don't yet have the biological markers that distinguish. There is some work ongoing in this field. I'm involved in one effort that looks at the individual biomarkers that distinguish one type of ALS from another type of ALS. There's some really exciting work going there, but it's not ready for prime time, so not ready for implementation into a trial as large as the platform trial. We ended up the session with what would be the ideal design of an outcome measure that we seek. Not a surprise, everybody wanted specific biomarkers that focused on different mechanisms relevant to that drug. We wanted a more direct measure of motor neuron decline. And of course, not surprisingly, there wasn't much disagreement about that. We all felt like we could do better, But at the moment, we're doing as best as we can. I guess one of the other advantages to being around for a while in the field is you can be a little bit more cynical, and having seen so much of excitement in a variety of drugs along the way, and also being really struck in my own practice about just how variable patients present, and how one patient can walk in and sometimes carry their portable ventilator, while the other one is much more debilitated from a limb involvement, but breathing is not as big of an issue. There's such tremendous clinical variability that I really do feel, and so did the panelists, that understanding that better was going to be very critical to advancing our understanding in better clinical trial design. So overall, I think it was, I forgot if I had another slide or not. These were the 3 participants that worked with me. Jeremy Scheffner, who is at Barrows Neurological Institute in Phoenix. Dr. Stacy Rudnicki, she works for Cytokinetics, and Bob Balo, who used to be up until very recently at Cedars-Sinai, is now with Roche in Switzerland. Each brought a very good perspective. I think the feedback from the attendees in the meeting was good so far, and it was for I think it was an interesting session that kind of gave us a reason to pause and look critically about our approaches at clinical trials. So I'd like to stop there. Hopefully I didn't run too far over the time, and be happy to answer any questions.
Thanks so much, Dr. Rosenfeld. No, you didn't run over time at all. Perfect timing, as a matter of fact. First question is around the relationship between ALS and PLS. Wondering whether drugs for ALS might have an effect on people who have PLS.
Well, it's a great question, and the answer is not as great as the question. The answer is definitely maybe. You know, on the one hand, PLS is a motor neuron disease. It's just that the motor neurons affected in the PLS patient are not the same motor neurons entirely as are affected in the ALS patient. Now, so on the one hand, you could say, yes, they're both motor neuron diseases. On the other hand, but the motor neuron diseases are different. So one school of thought is that motor nerve cells in general have only a finite way to die. There's only a handful of mechanisms that lead to cell death. There's more than one, but they're not infinite numbers. And perhaps if we could figure out how to address cell death in a generalized motor neuron disease, like we see in ALS, we will have a great foot up, a great understanding towards addressing the same problem in a more selective motor neuron decline in PLS. And for that matter, Parkinson's, Huntington's, ALS, Alzheimer's, all neurodegenerative diseases might actually share some common features. That's the most exciting prospect. The other reason why ALS is studied more than PLS, it's not a great reason, but the truth is that PLS patients progress much more slowly often. And as a result of that, finding a difference in a period of time is harder because they don't change as much naturally in that period of time. So if your drug makes a 20 or 30% change in the rate of decline, but you weren't declining that quickly to begin with, you may not be able to catch that. So they may be very related or they may be very different. Clearly, there's something susceptible about the upper motor neuron that is distinct from the susceptibility of the lower motor neuron, or everybody would have ALS. Whether that difference in susceptibility leads to or is related to the reason for the disease, that's a good question, and that we don't really know yet.
Okay, gotcha. That was a lot of great information. Thanks so much. We have a follow-up question to something that was asked a while ago. There was a question around, you know, knowing that veterans or military personnel tend to get ALS twice as often. Sort of as a follow-up to that, military aside, what are thought to be some of the causes of normal everyday civilian ALS? He's saying it'd be great to avoid those hazards.
Well, of course, that is the million-dollar question. And, you know, there have been some really wonderful epidemiologic investigations, which we thought we were onto something. I mean, I recall back earlier, some years back, there's a military base in San Antonio that led to a lot of publicity because there was a disproportionate number of people affected at that military base. And then they found out that most of them shared having worked in a particular building. And then there was articles that they actually worked on the same floor of that building. And we really thought this is all going to come down to a very specific toxin. There's been a lot of stories like that. I mean, there's endemic areas in the country, there's parts of the world, there's an Italian soccer team that had a disproportionate, there was a football team in the US. We thought perhaps it had to do with the chemicals on the field, treating the grass, the fertilizers and so on. The point is nothing really has been proven. And even though there is nothing proven, but certainly suggested, the takeaway message from that and many other studies is that clearly there is an environmental interaction. There's no question that there's some interaction, at least in parts, at least in some types of ALS, with the environment. There's a very established line of research that suggests it's a mold or an algae in certain parts. There's a toxin in another part of the world that seems to be. Now, it seems also likely that you need a certain genetic predisposition to have that toxin or that environmental agent affect you in a way to lead to ALS. Not everybody has that. So it's kind of a combination of perhaps a right genetic background, an exposure, a certain time, and it's the coalescence of these events together that we think gives you ALS. I don't think we've heard the last word on environmental exposures, and there's definitely interesting research going on right now about that, but I just don't think we have the final word on that yet.
Got it. Thank you. Another question. My mom is far along in her ALS and Parkinson's. She started symptoms in 2016, diagnosed 2017. She cannot move or speak, can only move her toes slightly. Are there treatments available for someone like her that would help her prolong her life?
Yeah, that's a hard question because if you're talking by treatments, this is one of those reasons, if you were in the audience, I could just ask you, but sorry, that if by treatments you mean clinical trials, generally speaking, clinical trials are limited to people where we have enough function to see an improvement, but enough dysfunction to see improvement as well. And so when you're at a certain point where you've lost so many motor neurons, if we were to provide back again 25% more or 30% more, would we be able to detect it? Chances are we might not. So I'm not aware myself of clinical trials that would be appropriate for somebody who is very late in the disease. However, there are many, many interventions that we try to improve the quality of life. It could be technology to help with movement. It could be speech, nutrition, ventilation. I mean, that's one of the focus areas of our program that I have in my clinics, which is that there is no stage of disease progression where we are not going to find some hopeful alternative to help you get by. Change the disease, take it away? No, unfortunately, we can't do that. But certainly we can try to make things better. And in that regard, yes, there is treatments. But if your question was referring to clinical trials, I'm not aware of that.
Okay. And the last question I'm seeing here, at least for now, what are the facilities that collect brain and tissue donation? It's important to have an accurate list because the process of donation must occur very quickly with a skilled team. I know locally in SoCal, UCLA is one place. I think they're just asking if there's a list or a place to find kind of all of them.
Yeah, that's a good question. I was thinking of that myself when the question came up before. That list changes from time to time because generally these are lab supported or funded projects that have a life of their own. I'm aware of the one at UCLA. I don't want to misspeak here because there's so many people listening in, but I will say that I can work with ALSA in trying to identify those places. I think that is a reasonable resource to have if that's of interest to you, and I will do my best to try to contribute to that. I just don't know. I used to be a site myself, but then we don't have that anymore. So I would say I will find out, and maybe with your help, Audra, we can post that with the resources of this discussion.
Sure, and I'm actually seeing a quick note from Dr. Finkbeiner that there's also one at UC San Francisco, and there are some efforts based at Mass General back east and in New York. So hopefully that helps answer that person's questions too. So let me see. I don't think we have other questions at this time. Thank you so much, Dr. Rosenfeld, for your time. Always a pleasure to have you be a part of this. And I just want to actually thank all of the panelists for all of these wonderful presentations. We're absolutely grateful to each of you for your time and commitment to both the Golden West chapter and to our ALS community. And thanks to all of you for your participation in all these important questions that you've asked today. If you have other questions on any of these topics, please do email askme@alsagoldenwest.org, and we will get some answers to your questions. Our programs are all made possible by individuals and families, foundations and companies who care deeply about the ALS community. And your generous support fuels absolutely everything we do, and everything we do advances the search for effective treatments and cures for ALS. And if you too would like to support the Golden West chapter, please consider making a donation today at give.alsagoldenwest.org. And please watch for the short survey on today's presentations that you'll receive via email in order to help us plan for future educational programs like this one. Thanks so much. Have a great day, and thanks for being here today.
Thank you, Audra, and to all the folks behind the scenes who made this webinar possible, and of course to all of our great presenters, Kuldeep, Steve, Clive, and Jeff. We couldn't do this without you, and it's an amazing contribution that we deeply appreciate. So thanks to everyone who made this possible, including the audience. Thank you.